Let's dive into the world of dendritic cells. These guys are like the intelligence agents of your immune system, constantly on patrol, always sniffing out danger, and then, bam, they prep for a targeted attack.
I like that, a targeted attack. That's a great way to put it. You see, dendritic cells are always on the lookout. Anything suspicious, and when they find something, they don't just sound the alarm, they gather intel, and they present it to the T and B cells.
Oh, so like the special forces.
Exactly, the specialized forces of your adaptive immune system.
So we're trying to understand how this whole thing works. How do these dendritic cells actually capture these antigens? What are the key receptors? And how does this all come together to shape the whole immune response?
Well, it's fascinating. We're learning so much about these cells. For example, we used to think that signaling pathways within cells were global.
Global?
Yeah, like affecting the whole cell equally. But research is showing that it's much more localized. It's happening in these specific little zones.
So not a citywide announcement, more like a targeted text message.
You got it. That's a great way to think about it.
But how does this all relate to dendritic cells?
Think of it like this. Those microdomains are like specialized departments within a cell, each one handling a specific task. And these dendritic cells, they've got a lot of departments to manage all those different signals they receive.
So if these microdomains are the departments, what are the workers?
That's where akinase anchoring proteins, or AKTs, come in. They act as scaffolds. They're keeping crucial signaling enzymes organized within these microdomains.
Like the office organizers, making sure everything is in the right place.
Precisely. And these AKPs, oh, they're especially important in dendritic cells, where they help orchestrate a precisely targeted immune response.
Okay, I'm starting to see the connections here. But before we get too deep into that, can we back up just a little bit? How do dendritic cells even capture those antigens in the first place?
Ugh, great question. It all starts with their impressive arsenal of receptors. They're covered in these receptors, each one designed to recognize specific patterns, specific motifs, the kind commonly found on pathogens.
So it's like they have a whole security team, each guard looking for a specific ID.
Exactly. And one of these key guards is a receptor called DC-cyan.
DC-cyan.
Yeah, and it's got, well, a bit of a sweet tooth. You see, it binds to certain sugar molecules.
Sugar molecules.
Yeah, the kind found on the surface of lots of viruses and bacteria.
So DC-cyan is checking for sugary treats, basically, as a sign of trouble.
That's a great way to put it. And when DC-cyan binds to its target, it triggers a whole chain of events within the dendritic cell, leading to antigen uptake and processing.
Okay, so the antigen is captured. Now what? How does the dendritic cell process this intel and prepare it for presentation to the T cells?
Ah, now that's where things get really interesting. The captured antigen is broken down into smaller pieces. It's like taking a document, shredding it into smaller bits of information. Then these antigen fragments, they get loaded onto special molecules called MHC molecules.
MHC. Does that familiar?
MHC stands for Major Histocompatibility Complex. And they're essentially presentation platforms for those antigen fragments. Think of them as like tiny billboards. They display snippets of the enemy's code for the T cells to analyze.
So the MHC molecules are like the couriers, delivering these key pieces of information to the T cells.
Exactly. And there are two main types of these MHC molecules, MHC class 1 and MHC class 2. They each have a specific role in shaping the immune response.
Okay, so MHC molecules are the delivery system. But how does the dendritic cell decide which class of MHC to use? Is it just random?
Not at all. It depends on where that antigen came from. MHC class 1 molecules usually present antigens that come from inside the cell, like a virus that infected the cell itself.
An internal security breach.
Yeah, like that. And this alerts a special type of T cell called a cytotoxic T cell. These guys are like assassins, programmed to eliminate infected cells. It's a pretty critical defense mechanism, especially against viruses and some types of cancers.
Wow, okay. That's amazing. So what about MHC class 2? What kind of intel are they presenting?
MHC class 2 specializes in presenting antigens that came from outside the cell. Stuff the dendritic cell engulfed and internalized, like bacteria, maybe some toxins.
So external threats.
You got it. And this information is then presented to helper T cells, which act more like commanders. They orchestrate the immune response by activating other immune cells, like B cells.
Wow, so it's like a really finely tuned communication system. The dendritic cell choosing the right MHC molecule to alert the right branch of the immune system.
Precisely. But there's another thing, a really fascinating aspect of this. Sometimes dendritic cells can do something called cross-presentation.
Cross-presentation with that.
It's a little bit of a rule breaker. Normally, those antigens from outside the cell are only presented on MHC class II molecules, which activates those helper T cells. But in cross-presentation, the dendritic cells, well, they can take those external antigens and present them on MHC class I molecules too.
So they're breaking protocol. Why would they do that?
It's a way to amplify the immune response. By presenting those external antigens on MHC class I, dendritic cells can activate those cytotoxic T cells, those assassins we talked about earlier. It's like adding an extra layer of defense against those threats that might slip through the cracks otherwise.
That's incredible, like calling in reinforcements.
Exactly. And this ability to cross-present, it's super important for fighting viruses and cancer cells because they often try to hide inside our own cells to avoid detection.
Like a way to outsmart those sneaky invaders.
Precisely. But even with all this sophisticated antigen presentation, there's one more really crucial element: co-stimulatory molecules.
Okay, now I'm really curious. What are these co-stimulatory molecules, and why are they so important?
Imagine a T cell, right? It encounters an antigen that's being presented on an MHC molecule. It's like getting a tip-off about a potential threat. But before it takes action, it needs some sort of confirmation. It needs a second signal to verify that, yes, this threat is real.
Like a double-check system.
Exactly. And that's where those co-stimulatory molecules come in. They're expressed on the surface of the dendritic cells, and they interact with corresponding receptors on those T cells. They provide that second signal.
It's like a green light.
Exactly. And without that co-stimulation, the T cell might become unresponsive, or it could even self-destruct.
Wow, it's like a really powerful safety mechanism, making sure that the immune system doesn't just overreact to harmless substances or even attack its own tissues.
Precisely. And here's where things get even more interesting. Dendritic cells can actually adjust the types and levels of these co-stimulatory molecules they express.
So they can fine-tune the immune response.
Exactly. It's like having a volume knob on the immune system's alarm system.
Wow. You're saying dendritic cells aren't just presenting the antigens, they're actually acting as instructors too, guiding the T cells towards specific destinies, like becoming those cytotoxic T cells or helper T cells.
That's exactly right. It's a remarkable testament to just how complex and sophisticated our immune systems really are.
This is mind-blowing. So how do scientists actually study these intricate processes? It's not like they can just sit down with a dendritic cell and have a chat, right?
Well, there are a range of really sophisticated techniques, and one of the most powerful is flow cytometry.
Flow cytometry.
Yeah, it's like a microscopic conveyor belt. Imagine that. Each cell is individually labeled, and they have these fluorescent tags that correspond to a specific molecule of interest. And then these labeled cells pass through a laser beam, and the light signals are detected and analyzed.
So it's like a high-tech barcode scanner for cells.
Precisely. It's a really powerful tool for studying the expression of receptors, those MHC molecules, and those co-stimulatory molecules on dendritic cells. It lets researchers really unravel the complexity of antigen presentation.
That's amazing.
Yeah.
But with all this talk about intricate pathways and molecular interactions, it's easy to kind of lose sight of the bigger picture. What does all of this actually mean for our health?
Well, understanding all these intricacies of antigen presentation, that's crucial for developing new vaccines and immunotherapies. By manipulating this process, we can boost immune responses against infections, cancers, even autoimmune diseases.
Wow, the implications are enormous. It's like unlocking the secrets of the immune system, gaining the power to really fine-tune its responses and harness its potential for all sorts of therapeutic purposes.
Exactly. And this brings us back to the importance of understanding those microdomains. They play a vital role in ensuring that the right messages are delivered at the right time to the right place.
Location, location, location. Just like real estate.
You got it. Within a dendritic cell, we might have different microdomains where specific receptors are clustered together.
Okay.
That allows for efficient signal processing, like a tailored immune response.
This concept of microdomains is really transforming how we understand cellular communication. It's like we've zoomed in from a satellite view of a city down to street level, and we can see all those intricate interactions, all the networks that make everything work.
Precisely. That level of detail is crucial for understanding the nuances of antigen presentation. Dendritic cells are constantly sampling their environment, taking up antigens, processing them, and getting ready to present them to those T cells.
Okay, but there's so many potential antigens out there. How do dendritic cells even know which ones to focus on? It's like finding a needle in a haystack.
Ah, that's where those specialized receptors come in. Dendritic cells are equipped with this whole arsenal of receptors that recognize specific patterns or motifs that are common on pathogens. Think of them like molecular fingerprints, allowing them to tell friend from foe.
So those receptors are like the security guards at the entrance to the immune system headquarters, checking IDs and flagging any suspicious characters.
Exactly. And this brings us back to DCSIGN, remember, our sugar-loving detective.
Always on the lookout for those sweet treats that might betray an invader.
Precisely. And when DCSIGN binds to its target, it triggers this whole cascade of events within the dendritic cell, leading to antigen uptake and processing.
Okay, so DCIGN's got a key role in capturing those external antigens. But then what happens to the antigen once it's inside the dendritic cell? It's not like it can just walk into the T-cell meeting room and announce itself.
That's where those MHC molecules come in. Remember those tiny billboards we talked about?
The ones that display the enemy's code
Yeah.
for the T-cells to analyze.
Exactly. They act as presentation platforms, showing those fragments of the captured antigen to the T cells. And we talked about the two main types, MHC class 1 and MHC class 2. Each one has a specific role.
MHC class 1 for internal threats, right, to alert those cytotoxic T-cells, those assassins.
You got it. And MHC class 2, that's for those external threats. It presents those antigens to the helper T cells, the commanders of the immune response.
Okay, yeah, it's all coming back to me. But you mentioned something really interesting, cross-presentation, right? Remind me about that again.
Of course. So cross-presentation is when dendritic cells kind of break the traditional rules and they present those external antigens on those MHC class 1 molecules.
So those external threats get shown to the cytotoxic T cells, those assassins.
Exactly. It's a great strategy to amplify that immune response, add an extra layer of defense against invaders that try to hide.
So it's like the dendritic cells are saying, we're not taking any chances. Alert both branches of the immune system just to be safe.
That's a great way to put it. And they're incredibly versatile, constantly evolving.
So we've talked about the antigen capture, the processing, and the presentation. But what about those costimulatory molecules again?
Oh, yeah. Those co-stimulatory molecules, they provide that second signal, that confirmation that the threat's real.
It's the green light.
You got it. And remember, they can fine-tune that immune response. It's like having that volume knob on the immune system's alarm system.
So they're not just presenting the antigen, they're controlling the intensity of the response, too.
Exactly. They're like skilled conductors.
It seems like every time we peel back a layer, we just find even more complexity.
That's the beauty of it. And it brings us back to the importance of understanding those microdomains, remember?
Right. Those specialized departments within the cell.
Precisely. And it turns out the location of the antigen presentation within those microdomains can actually influence the fate of the T cells.
So where the antigen is presented matters just as much as what's presented.
Exactly. It's like having different meeting rooms within a company. The location of the meeting might actually influence what happens in the discussion.
That's a great analogy. So how does the dendritic cell decide where to present the antigen in these microdomains?
That's a tough question. Scientists are still working on all the details. But it's clear that the signals the dendritic cell receives from its surroundings play a crucial role.
So those chemical messages, the cytokines, chemokines, PMPs, all that.
Exactly. Those signals influence the expression, the localization of the MHC molecules, the co-stimulatory molecules, and ultimately, they shape the entire immune response.
This is mind-boggling. It's like the immune system's this dynamic, responsive network that's constantly adapting.
Absolutely. And those dendritic cells are right at the heart of it all.
This has been an incredible deep dive so far. But before we move on, can we talk about the different types of dendritic cells? You mentioned conventional dendritic cells, cDCs, and plasmacytoid dendritic cells, pDCs.
Yes. Understanding the differences between those subtypes is really crucial for understanding how the immune response works.
Okay, I'm ready. Tell me about these cDCs and pDCs. What makes them unique?
Well, cDCs, they're the ones we've been talking about mostly. They're like the classic antigen-presenting cells. They're always patrolling the tissues, capturing antigens, and they migrate to the lymph nodes to activate those T cells.
So they're the frontline soldiers, gathering intel, alerting the immune system.
Precisely. And now pDCs, they're more specialized. They're really good at recognizing viral infections, and they produce tons of interferon.
Interferon, that's one of those cytokines, right?
That's right. Interferon's a powerful signaling molecule. It helps control viral infections by, well, interfering with viral replication. It also activates other immune cells.
So pDCs are the virus-busting specialists. They release this weapon that disrupts the viral replication and rallies the troops.
That's a fantastic analogy, and that specialized function, it's essential for controlling viral infections, especially in the early stages.
This is fascinating. So the immune system has this division of labor, different cell types specializing in specific tasks: antigen presentation, virus detection, interferon production.
Exactly. And that specialization allows for a much more efficient and targeted immune response.
Okay, so we've got cDCs, the generalists, and pDCs, the viral specialists. But are there more specialized subtypes within the cDC family trait?
Absolutely. cDCs can be further divided based on their location, their function, the types of receptors they've got. For example, there are Langerhans cells. They live in the skin, act as sentinels, detecting those inviting pathogens.
Border patrol of the immune system.
Exactly. And then you've got dermal dendritic cells residing in the deeper layers of the skin. They're involved in wound healing and responding to allergens.
Yeah, having different layers of security.
You got it. And on top of those skin-resident dendritic cells, there are others that circulate in the blood and the lymphatic system, constantly checking our tissues.
It's a complex network. Dendritic cells are everywhere.
They are. And their ability to specialize and adapt to all these different environments, that's what makes them so important for a robust and effective immune response.
This is incredible. We could probably talk for hours just about the different types of dendritic cells.
Oh, absolutely. But for now, maybe let's shift gears back to the antigen presentation process itself. There's still so much more to explore.
All right, I'm ready. Lead the way.
You know, it's incredible how dendritic cells can fine-tune the immune response by adjusting the types and levels of co-stimulatory molecules they express.
That's like they have a control panel, dialing the response up or down depending on what's happening.
Precisely. And that fine-tuning, it's crucial for preventing things like too much inflammation, which could damage tissues, but also, immune suppression, that could leave us vulnerable to infection.
It's a balancing act.
Yeah.
But how do they make those decisions? What tells them which co-stimulatory molecules to use?
It's all about the signals. They get signals from their surroundings. They're really sensitive to things like cytokines, chemokines, and those PMPs we talked about before.
Oh, right. Those cytokines, chemokines, they're like the chemical messengers.
Yeah.
Little notes pass between cells.
You got it. And those PMPs, they're the alarm signals released by pathogens or damaged cells, red flags.
So it's a communication network. They're getting info from other immune cells and indeed of the invaders.
Exactly. It's a constant flow, letting them tailor their response to the specific challenge.
Can you give me an example, like how this fine-tuning actually works?
Sure. Let's take a viral infection. When a virus gets into a cell, it releases PMPs, like viral RNA or DNA.
So it's leaving traces.
Yeah, and those PMPs, they trigger pattern recognition receptors, PRRs, on dendritic cells.
So those PRRs are like sensors.
Exactly. They pick up those telltale signs of the virus, and when those PRRs get activated, it sets off a whole chain reaction inside the dendritic cell. Co-stimulatory molecules get upregulated, like CD80 and CD86.
So it's like, hold on, viral situation here.
Exactly. And those upregulated molecules, they provide that second signal, the one that activates the T cells.
Okay, that makes sense. We've talked a lot about T cells. What about B cells? Where do they fit in?
These cells are really interesting. They can actually present antigens themselves, even though their main job is making antibodies.
Really? I thought that was mainly dendritic cells and macrophages.
Oh, they're definitely the stars. But B cells can help out too.
Okay, I'm all ears. Tell me about this.
So B cells have these special receptors called B cell receptors, or BCRs. They're like antibodies, but customized. Each one recognizes a specific antigen.
So each B cell has its own specialty.
Exactly. And when a BCR binds to its matching antigen, well, the B cell takes it in, breaks it down.
Like dendritic cells do.
Just like that. Then those little antigen pieces, they get loaded onto MHC class II molecules, and they're presented on the B cell's surface.
So they can present to helper T cells.
Exactly. And that interaction, that's what helps fine-tune the antibody response.
How so?
While those helper T cells give signals, they help the B cells do a few things: proliferate, differentiate, and then make those really specific antibodies, the ones that target the pathogen.
It's like a team effort.
Yeah.
B cell shows the antigen, T cell gives the instructions, and then boom, antibody army.
It really is beautiful collaboration.
This is amazing. The immune system is so much more complex than I thought.
It is, and the more we learn about these interactions, the better we'll get at developing new treatments for all sorts of diseases.
I'm definitely inspired to keep learning. But one more question. We've talked about how dendritic cells capture and present, how they fine-tune, how they work with B cells, but where is all this happening?
Good question. It's all happening in this amazing network of tissues and organs. The lymphatic system.
Lymphatic system, I remember that name, but refresh my memory.
Think of it like this. A huge network. Vessels, nodes, organs. It's the immune system's surveillance and communication hub.
Okay, I can kind of picture it, but how does it actually work?
Those lymphatic vessels, they're like a separate highway system. Running right alongside your blood vessels, they gather fluid from all over the body and carry it to the lymph nodes.
Lymph nodes. Those are the little bean-shaped things that get swollen when you're sick, right?
Exactly. They're everywhere, acting as filters and checkpoints, packed with immune cells, dendritic cells, T cells, B cells.
Meeting points for the immune system.
You got it. When dendritic cells grab those antigens out in the tissues, they travel through those lymphatic vessels to the nearest lymph node.
Like couriers bringing the news.
Perfect analogy. And once they're there, they present the antigens to the T cells, and that kicks off the immune response.
It's like a spy movie. The dendritic cells are undercover, getting the info to headquarters, and then the special forces get deployed.
I love that. And that ability to move around, to communicate between different parts of the body, that's what keeps the immune response coordinated and effective.
Mind-blowing. The lymphatic system is like the unsung hero here.
It really is. Without it, our defenses wouldn't stand a chance.
I'm definitely adding it to my list of things to be thankful for. But those dendritic cells traveling to the lymph nodes, how do they find their way?
That's where chemotaxis comes in.
Chemotaxis. Now that sounds familiar.
Think of it like a scent trail. Cells release these chemical signals called chemokines, and they attract other cells, guide them to where they need to be.
So it's like a trail of breadcrumbs leading to the lymph nodes.
You got it. Lymph nodes release those chemokines that attract dendritic cells. That way, they know where to go to present those antigens and get the response going.
It's amazing how much communication is going on, all at the microscopic level.
It really is. And all that movement, all that communication, that's what keeps the immune response balanced and healthy.
This is fascinating. But what if a T cell doesn't find its antigen in the lymph node? Does it just keep searching forever?
Not necessarily. T cells don't live forever. If they don't find their match, they go through apoptosis.
Apoptosis, that sounds kind of scary.
It's programmed cell death. It's a natural process, gets rid of cells that aren't needed anymore, or maybe they're damaged.
So it's like quality control.
Exactly. Keeps the system running smoothly, and all that turnover. It helps keep the immune system diverse, ready for anything.
It's incredible. Always adapting, even at the individual cell level.
That's the beauty of it. It's dynamic. That's what makes it so effective. We live in a world full of pathogens and threats, and it's always changing.
This has been amazing. We've gone from the tiny details of antigen presentation to the lymph nodes and these cells moving all over the place. It's so complex.
And coordinated. That's the key. It really shows the power of nature.
So we've talked about the antigen capture, the activation, the T cells. But what happens when a T cell actually finds its match? What happens then?
That's when things get really exciting. Once that T cell finds its matching antigen and it gets those costimulatory signals, it's like a switch flips.
Go time.
Exactly. It undergoes this huge transformation. It starts dividing like crazy, creates a whole army of clones.
So many copies, all with the same antigen specificity.
You got it. And they differentiate too, become these specialized subtypes, each with its own job to do in the fight.
Oh, right. We talked about those different types of T cells before.
Yeah, those cytotoxic T cells and the helper T cells.
The assassins and the commanders.
Exactly. And depending on what the threat is, what signals they're getting, the activated T cells can become either of those or even other types, like regulatory T cells. They help keep the immune response in check so it doesn't go too far.
So it's like a choose-your-own-adventure.
Yeah.
Depending on the signals.
That's a great way to put it. And that flexibility, that's key for a balanced, effective response.
Okay, so now we've got these activated T cells ready to fight. But how do they actually get rid of the threat? What are their weapons?
Depends on the type of T cell. Cytotoxic T cells, while they're really good at killing infected cells, cancer cells, they release these toxic molecules.
Toxic molecule.
Yeah, they punch holes in the target cell's membrane, and that triggers apoptosis.
Oh, right. Programmed cell death. It's like a clean getaway.
Precisely. No collateral damage. Now, those helper T cells, they don't do the killing directly, but they're really important for coordinating everything.
Right, they're the commanders.
Exactly. They release all these cytokines, they activate other immune cells, like macrophages and B cells, help them fight better.
So they're giving the orders.
You got it. And it's that coordinated attack, all the players working together, that clears the infection, gets the body back in balance.
It's like watching a perfectly choreographed dance, but it's a dance of destruction.
I like that. And it really shows the power of the immune system. All this coordination, constantly adapting, evolving.
We've gone from the capture of the antigens to the activation of the T cells to the elimination of the threat. We've seen how the antigens are presented, what happens in the lymph nodes, how those lymphocytes travel. It's just amazing, all this complexity, how well it all works together.
It really is. And the more we learn, the more we realize there's so much more to learn. There's so many possibilities, so many potential therapies, treatments.
I'm leaving today with this whole new appreciation for my immune system. It's doing all this incredible work all the time, and I don't even realize it.
That's right. And as we keep exploring, we're not just understanding it better, we're also opening up new ways to improve health well-being.
This has been such an incredible deep dive. Thanks for joining us, and we hope you learned something new about antigen presentation and dendritic cells.
Keep exploring. The journey never ends.